Hello, everyone, and welcome to our Research Participant Symposium in honor of Stephen D. Johanson. We had hoped to have this event in person, but given the circumstances, we decided to hold a virtual event so we didn't miss the opportunity to honor your dedication to making our research possible. But before we get started, I want to help you navigate the virtual format that we're going to be using. So you will be able to participate in this event by asking questions or making comments, but you need to do it in that Q&A box. You see that red arrow there? So just click on that, then type in your questions and comments, and then later on during the panel, we will answer your questions and state your comments. So that's how you're going to interact with us. All of you who were invited to this webinar participate in one of the many affiliated research projects across the Massachusetts Alzheimer's Disease Research Center, or affectionately called MADRC. The MADRC spans both the Massachusetts General Hospital and Brigham and Women's Hospital, and as the picture indicates, we want you to feel that we are your research home. Some of you are in clinical trials to test drugs to help find a cure. Others are involved in observational research where you help us develop new diagnostic and imaging procedures and therapies. Whatever research study you are in, you are a research hero to us. We have an exciting program this morning, but first I want to introduce you to all the research teams who would love to say hi and reach out to thank you. So first we have all the people who take part in the Harvard Aging Brain Study, which is under the direction of Drs. Risa Sperling and Keith Johnson. You will be hearing from Dr. Sperling in a few minutes, but we want you to know that we have been renewed for an additional 5 years, making this a total of 15 years of funded research, all because of you. We cannot be more grateful for your dedication. And then there's a large number of you on this call today that are a part of the memory study, which is under the leadership of Dr. Teresa Gomez-Isla and Deborah Blacker in the gerontology unit at the Charlestown Navy Yard. A number of you have participated in this study for many, many years and have helped us contribute to the national effort to understand aging and Alzheimer's disease. We also have the clinical trial arms of our MADRC Center. First, the Center for Alzheimer Research and Treatment, which is at the Brigham and Women's Hospital and is under the medical direction of Dr. Gad Marshall. At CART, we sponsor various prevention trials and drug studies to help treat Alzheimer's disease from its earliest to its latest stages. And new to the clinical trial efforts at MGH is the Alzheimer Clinical and Translational Research Unit, which is under the direction of Dr. Steve Arnold. The ACT crew, which we sometimes coin it, performs proof of concept studies to help find novel cures for Alzheimer's disease, Lewy body disease, and frontotemporal dementia. And finally, where does all that data go? Our MADRC Data Core, led by Dr. Sudeshna Das and Lori Chibnik, work behind the scenes, taking the data we collect from you, coordinate it with other data centers, both locally and nationally, and store it safely so that we can analyze it later. And last but not least, the organizers of this event, as well as your Outreach, Recruitment, and Engagement Corps, also say thank you to each of you for attending. So this year, we are honoring one of our research participants who sadly passed away from early onset Alzheimer's disease in 2018. Stephen D. Johanson was Stephen D. Johanson. And I want you to meet Stephen's wife, Judy, who will tell you more about him and help put this event in perspective. So Judy?
Thank you, Dr. Rentz, and I'd like to echo your words of welcome. Our family is humbled and honored to have Steve's name attached to this inspirational event and to be in virtual presence of so many MADRC family today. Our daughter-in-law, Sarah, created this collage of Steve that you might gain some insight into the man attached to the name. Steve was a visionary of sorts. He grew up here in Boston, but could always see beyond the boundaries of the city to the mountains and oceans off in the distance. He loved to travel and explore, but he also embraced the comfort of home. Steve was a lover of nature, an avid reader, a closet writer, and a recreational sailor, and so much more. He was a dreamer and a doer with boundless energy to make the world a better place. He found beauty in each day and embraced life with joy and wonder. When we met, we fell instantly in love and began our fairy tale story, complete with 2 children, a home, a fulfilling life, and eventually grandchildren. He was the most supportive partner and a father with endless patience. He was my home and I was his. Together, we had it all. We seldom tired of each other's company, and we're so grateful to be able to walk life's journey together, hand in hand. We welcomed each new season of life and found resiliency to be our friend as we watched our kids go off to college, and then both of them falling in love themselves and getting married. The endowment of grandchildren was proof that our lineage would continue and carry our name and DNA into future generations. Our story would go on. Where 2 once walked, now 9 of us collectively trod on this well-worn path of family. Steve had a talent for envisioning structures not yet built. He was an engineer by nature and trade. He utilized his skills around our home and in his career as a construction manager for the Special Projects Division at Northeastern University. It was in this role that he began to struggle with the tasks associated with his projects in 2011. 5 months of testing eventually led us to a diagnosis of younger onset Alzheimer's at age 59 and another season of life to adapt to. We knew then that our time would be measured in the quality of our experiences as opposed to the quantity of years. So we held each other's hands tighter and began walking this new road. The properties of what mattered most in life to Steve caused him to participate in research. For our children and their children's sake, he was compelled to take action. He knew that the only path to a treatment, cure, and prevention of Alzheimer's and other related dementias was through research. Our research visits offered him empowerment in the midst of his disease. He was fascinated and appreciative of the brilliant and compassionate people who are working so hard to figure this all out. He couldn't change his diagnosis, and he knew he would not be around to teach our grandchildren how to sail. But by actively engaging in research, he has left a legacy that we are all so proud of. He was a husband, a father, a grandfather, and a friend. He was a passionate research participant. He was a visionary. Steve left his brain to the Mass Alzheimer's Disease Research Center and his heart to me. I'm blessed to keep them together as we work towards that structure of hope that he envisioned through the power of research. On behalf of our family, I want to thank each and every one of you, because in my eyes, you are all visionaries. Your active participation in research will indeed change the world. Because of you, one day we will tell a future generation that there used to be an incurable disease called dementia, and how many people laced up their boots, joined forces, and tamped down a path to a cure. And because of you, that story will never be forgotten. Thank you so much.
Judy, thank you. The doing your pad toward research. I'd like to now introduce you to Nancy Koppelman, our Director of Community Engagement, who's going to introduce you to a very special guest this morning. Nancy?
Thank you, Doreen. And I am delighted to introduce Dennis Chan, who is a research participant and research ambassador at the Center for Alzheimer Research and Treatment. Dennis was just awarded the 2020 Global Alzheimer's Platform Citizen Scientist Catalyst Award. The awardee acts as a stimulus in bringing about or hastening a result through creative, new, and novel approaches to encourage clinical trial participation. As a new member of the Alzheimer's Clinical Trial Consortium Advisory Board, Dennis attended the meeting at the University of Southern California, San Diego, earlier this year, and continues to talk about the importance of participating in memory research whenever possible. Dennis?
And I have been in the A4 study for about 3 years now, and our experience has been much more positive and rewarding than I had imagined. Like a lot of people, our family experienced the effect of dementia. Angie's maternal grandfather, paternal grandmother, and 2 uncles all died of Alzheimer's disease. My mother had Parkinson's and developed vascular dementia after her strokes. About 10 years ago, Angie developed significant memory difficulties at age 55. It was very scary and stressful for the entire family. Finding an explanation for her symptoms was difficult and frustrating. After almost 3 years and numerous doctor visits and tests, she was still without a diagnosis. We finally figured out the symptoms were side effects of her medications. Fortunately, her symptoms improved after stopping the meds. During this episode, we received much needed support from the Alzheimer's Association and learned a lot about the disease. We became aware of trial match, but it took a few more years before I became a study participant. So, what made me finally consider participation in Alzheimer research? I've always admired the courage and dedication of doctors and nurses who go on Doctors Without Borders assignments in the most dangerous places on earth to help people most in need of medical care. One day, in a family conversation, one of my kids challenged me and asked, "Why don't you do something about it instead of just writing checks?" It made me realize I could do more. Donating money is good, but not quite the same as participating in the fight. I cannot be a field doctor or nurse, but I can participate in a clinical trial to help find a cure for Alzheimer's. I also want to acknowledge some perks that enhance my experience of being a research participant. With Angie's family history and my lack of symptoms, we were both quite surprised when I qualified for the A4 studies instead of her. Although I'm preclinical, it is good to know my condition is being followed and I will have access to quality care if and when I need it. The CAT staff are extremely friendly, caring, and supportive. I just wish all doctor offices were like that. The CAT support group for participants and partners is a great place to meet like-minded people who are at different stages of the disease and are dealing with the ups and downs of the condition. We share resources and learn how one another deal with different situations. Moving a meeting to Zoom when the studies were interrupted by the pandemic was a great idea. It kept the group together and also in touch with the happenings in the clinic. Taking part in the ACTC Research Participant Advisory Board was another great experience. They allow me to contribute feedback and input through the lens of a study participant to the design of future studies. Thanks to ACTC, I was able to attend the virtual Alzheimer's Association International Conference and also other events to learn more about the disease and the current state of research. I became aware of the importance of diversity in clinical study participants, and that past injustice and the current economic and healthcare inequity are keeping Black and brown people away from participating. Being Asian, I'm a member of a minority group, so I hope my story will encourage more people, especially people from underserved communities, to participate in clinical studies. Most rewarding of all, I feel like I'm part of a team working on a common goal to improve the life of many. Thank you.
Thank you, Dennis. And thank you so much for everything that you've done. What a beautiful talk. We really appreciate it. Now, there's a special message that comes from Anthony Everett. Some of you may know him as the Emmy Award-winning anchor of Chronicle, and he wanted to speak to our research participants as well about the importance of participation. So let's listen to him.
Hello, everyone. My name is Anthony Everett. I am the host of Chronicle, the long-running television news magazine of WCVB-TV, and I am very honored to be here with you today. I come to you this morning from a couple of different perspectives. One, of course, is a journalist who has covered the scourge of Alzheimer's for a number of years and from a number of perspectives. We certainly have spoken with doctors and scientists, researchers, and research participants like yourselves, all of whom are working toward a common goal of understanding Alzheimer's and developing both therapies and ultimately, of course, a cure. I speak with you as well as a friend who has witnessed the devastation up close that Alzheimer's has inflicted on some of my good friends and colleagues. And I also speak to you as the son of a man who battled Alzheimer's, whose final years really were taken from us, from him, as he too battled that slow robbery of the mind that took him away from us and his connection to the world around him. So I speak with you from these various perspectives, but the message that I have for you today is really singular, and that is one of gratitude. Just gratitude. The scientists, the doctors, the researchers who are working every day to understand and unravel Alzheimer's are heroic, to be sure. But many of them have told me very specifically themselves that none of their efforts are possible without you. You, who donate your time and your energy to clinical trials, observational studies. It is that participation that makes a difference and will make a difference in advancing the research, in moving the front line forward, if you will, in the battle against Alzheimer's. If anything, we need more courageous research participants like yourselves. Finding enough research participants and those that mirror the population in general has been difficult. In the best of times, it is even more so now as we are experiencing, of course, this global pandemic. You are incredibly important to the effort to find therapies and a cure. I'd also like to thank Judy Johanson for reaching out and extending the invitation to speak with you today. And this is a wonderful event that is, of course, named in honor of her late husband, Stephen, and the battle he endured with Alzheimer's. So thank you, Judy, for that. As my good friend Greg O'Brien, I know many of you know him, often says, Alzheimer's is a family disease. A diagnosis of one affects many. I know that, you know that, Judy knows that. What Alzheimer's doesn't know is that the strength of family ultimately will carry the day. Your family, this family of good people working together for a common cause. So from myself and from my family to this incredible group of research participants, I simply say thank you. Keep up the good work. we will find a way and a path back from the darkness and to therapies and to a cure. I am sure we will do that, but we will never get there without you. So thank you for inviting me today. Thank you for your hard work. Have a good day. Be safe, stay well, and keep up the good fight.
Wow. You know, it's between Dennis and Anthony and Judy, very inspirational. And but we want you to know that it is because of all of you that you make our work possible. I do now want to move on to why we're here. It's my pleasure to introduce to you the director of the Massachusetts Alzheimer's Disease Research Center, Dr. Brad Hyman. Dr. Hyman is the John P. Penny Professor of Neurology at Harvard Medical School and a world leader in understanding the basic proteins involved in Alzheimer's disease. He's going to speak to us today about current developments in research to move us closer to a cure. Brad?
Thank you, Doreen. Doreen, can you see the slides now?
Yes.
Okay,
Perfect.
super. Well, first, I just want to say, welcome and thank you. I've been watching the numbers in the bottom of the screen go, and we have over 150 participants. That's really amazing. If I think about the auditoriums at the Mass General, I don't think we have a single auditorium that would hold 150 people. And now we have 150 of us gathered together for this single purpose, to celebrate the contributions that you all have made and to let you know what we've been up to, trying to keep working against this awful, awful disease. And I especially want to thank Doreen and Judy for organizing this, and a beautiful tribute to Stephen, who I had the honor of knowing, of course, and Dennis and his moving words. Really, what an amazing group of people to come together. So from my perspective, what I'd like to do in just a few minutes is think about what we know now about the diseases of aging that affect cognition, what we need to learn together, and what we can together contribute to lead to a cure. Okay. The best place to start, of course, is with our participants and the courage and energy that you all bring. And no one epitomizes that better than Judy and her family. It's just a wonderful picture for some years ago, family bands together to fight Alzheimer's. And I think of all the participants in the Alzheimer's Center and the affiliated studies that as one big family, and we take care of each other and we help each other. And I hope that you see that we are reaching out to you to not just today, but to always be there for you. What we all want to do at the end is to find a way to cure these diseases. And that's been a frustratingly difficult task. We've been at this for 30 years. I moved to Mass General in 1989, so a long time ago. And every few years, a scientist or a doctor will be asked on TV, well, how long will it be till we have a cure? And now I'm happy to tell you that having observed the field for over 30 years, I know the right answer to that question, because every time I've ever heard anyone ask that question, they say, well, we hope within 5 years. And so the right answer is within 5 years. I hope that, in fact, finally today, that's right, because the progress that we've made over the last decade has really been extraordinary. And I want to share some of that with you, and also share with you some of the things that we've learned that really send us in a slightly different direction than maybe we were before. So I'm going to start by asking the questions. What causes are there to developing dementia? Do different people have different reasons that they develop dementia? And how could we tell, if people have different reasons, what the right medicines for them might be? And one of the things that we've learned really from you over the years is that everybody's different. And understanding those differences, I believe, is going to be one of the absolute keys to understanding how to cure this disease. One of the reasons that the fact that everybody's different is important is that many of the clinical trials that we've run over the years makes the assumption that people are more or less the same. And yet, what we've learned really is that everybody's different. And what we want to learn, and the things that our trials, our observational trials, our clinical trials are really drilling down on now, is how to tell the small ways in which everyone is different and the big ways in which everyone is the same, and sort out those two things. I'm going to just show you some data from our own studies. These are participants over the last 20 years in our Alzheimer's studies here at Mass General and the Brigham. On the left of the slide is sort of a cartoon version of how we all think that a drug study might work. Is that we would take patients, and if we gave them a sugar pill, a placebo, if we essentially didn't give them an effective medication, that they would progress. On average, they'd progress by a little bit each year, and over the course of 2 or 3 years, they'd progress by a known amount that we could predict. And if we gave them the medication, the best of all cases, they would get better, but at the very least, we would hope that the medication would differentiate from the placebo group and improve things, shown here by moving down the line in the rate of progression. That's the cartoon version. That's what we sort of had in our minds when we were designing trials a decade ago. But when we look at the actual data, and when we look at individuals whom we see, month after month and year after year, we find out that idealized version of everybody progressing at the same rate really isn't right. And this is true in my experience as a clinician. It's true in everyday life, just seeing friends and colleagues and coworkers begin to be affected by these diseases. And it turns out that everybody's different, that some people sadly begin to develop the illness, and then just over the course of a very short period of time, over 2 or 3 years, really progress in a dramatic and awful way. And other people, although over the course of years, no doubt they're getting a little bit worse, they gotta kind of hang in there. They're resilient to the disease in some ways. And if you look at a group of about 30 people on the right-hand side, you can see that these lines show that some people are really fast, some people are really slow. And if you average all those people together, what you get is sort of an average, but it doesn't tell you very much about how any individual would progress over time. And this is one of the fundamental problems in trying to tell whether or not a drug worked. Because if some people are going to do fairly well, no matter what happens, the drug doesn't help them that much. And if some people are going to do miserable, but are due to a different driver than your drug is affecting, they're going to add noise too. And so we need to learn what's different between the individuals who have a really, really rough time of it and the individuals who seem to be relatively spared. And I'll add that this is true not just of Alzheimer's disease, but it's hard to even look at a newspaper today and not see COVID and think about how some people with COVID end up in the intensive care unit and sadly pass away, and other people are completely asymptomatic. The same thing is true in cancer. So throughout medicine, we see this sort of individual differences in how people respond to disease processes. And this is one of our goals. And this is why every single one of you who's participating is adding valuable data to the study. So one of our goals is to understand how people differ from one another and how they're the same. So what are the questions you're helping with? Why do some people develop memory problems and other people don't? You know, we heard from Dennis Chan. He qualified for the A4 study. That means that there's some amyloid that's in the brain, but he's doing great. Other people with amyloid in their brain aren't doing great. What's different between them? And then, of course, why do some people get worse quickly and others don't? So in the Alzheimer Center, and this is mirrored by HABS and the other research teams, there are clinical units in which neurologists and psychiatrists and geriatricians perform examinations. We do the memory tests. We obtain blood samples so that the laboratories that are associated with the studies can look for biochemical markers of disease progression. Sometimes we'll look in the blood. More often and incredibly valuably, we'll ask participants to have a spinal tap so that we can examine the fluid that actually bathes the brain. It makes perfect sense that the fluid that's bathing the brain would reflect what's going on in the brain to a greater extent than the fluid in your blood, in your arteries and veins, which are going everywhere in the body. And so the combination of plasma and spinal fluid, CSF, is so invaluable. And we thank you, those people who were really brave enough to undergo a lumbar puncture to help with the research effort. We have a whole team that's advancing neuroimaging, MRI scans and PET scans, and some of the major advances in the entire world on PET scans with the amyloid scans and the tau scans were made right here in Boston by our team of investigators. The neuropath core is absolutely critical. You know, we talked to each of our participants about the importance of autopsy, and Judy mentioned that Stephen had undergone an autopsy. That tissue provides the absolute truth about what's going on with different individuals, and we've learned so much from it in the past years. The AuraCore, with Doreen leading it, communicates to our participants, recruits people for clinical trials, keeps everybody on the same page. Is so important. And a shout out also to an educational component in the centers in which our young investigators, the new fellows, the medical students, and so forth, are trained in how to care for patients and trained in how to do the science to keep going strong in future years. So what are the next things we're going to be asking you to help with? Well, I sort of mentioned the blood and the spinal fluid. We're going to ask whether or not they predict an individual's course, so that every time you come back to the clinic and are tested again, that adds more information to these ongoing trials. We're learning what changes occur in the brain, those that we can see on an MRI scan or a PET scan, and those that we can see under the microscope at autopsy. And they can help tell us which persons had problems with cognition and which didn't. There's this whole group of resilient individuals whose brains have plaques and tangles, but who are cognitively okay. And we're beginning to learn what's different in those brains. It looks as if there's a class of inflammatory molecules that occur in one instance and not in the other. And we're trying to understand if that's really a guide to therapy. So what are the challenges? What are the biological, social, environmental factors that contribute to risk for dementia or rapidity of progression? And so a lot of the questions that we ask you about what other medications you're taking, about whether or not you're sleeping okay, about where you live, about whether or not you have high blood pressure, that's all feeding into the statistical models that our team is building to try to understand the whole picture, the whole body, the entire process of what's going on. When we ask you to participate in affiliated studies that are focused on biomarkers in the spinal fluid or the plasma, on imaging, on PET scans or MRI scans, on better and more refined and robust clinical tests, even in the past couple of years, things that I'm very excited about with digital markers in which we ask you to fill out things on an iPad or look at a computer and we track how your eyes are moving. All of those things are critical and go into the big picture of telling how one individual is different from another individual. And ultimately, I hope that we can answer the question for any given intervention, how to choose those patients who are most likely to benefit from it and determine how to tell where it would be most effective. So looking towards the next 5 years, I just want to think about your future, about whatever your future holds. And thank you for going there together. And thank you really on behalf of our entire team here at the center, with special thanks, of course, to Judy Johanson there in the middle and Doreen for organizing this. Really appreciate your coming and joining us today. Thank you.
Thank you, Brad, very much. It's inspiring to know that there is a path forward and that in 5 years, we're going to find a cure. It's now my pleasure to move on to our next keynote speaker, Dr. Risa Sperling. Many of you know that Dr. Sperling is a leader in developing prevention trials for Alzheimer's disease. She's a professor of neurology at Harvard Medical School and the Redmond Family Distinguished Chair of Neurology at the Brigham and Women's Hospital. Today, she's going to speak to us about current developments in clinical trials, how together we are fighting for a cure. Risa?
Thank you so much, Doreen. Let me first make sure that my slides are showing up okay. Does that look?
They look great.
Great. Okay. So first, I really want to thank you for organizing, and I want to thank everybody who's joined us today, because this symposium is really about you and about how much you are helping us find answers. So I first especially want to thank Judy and Steve. I had the pleasure of meeting them at a outreach event, I think more than a decade ago. And also apologize that I'm gonna have to leave right after this because I have seen Alzheimer's up and close and personal. Unfortunately, it took my dad a couple of years ago, and I need to leave to take care of my mom who's having surgery. So I apologize for not being here for later for questions. But I have the honor today of really talking about how your contributions to our research has really helping us move forward in clinical trials. And in particular, I'm here today representing the Harvard Aging Brain Study and how some of the findings in the Harvard Aging Brain Study have really informed the clinical trials, and in particular the prevention trials, but other studies as well that we're running in our clinical trial units, including CART. So I think many of you have seen a slide like this before, but I do think one of the first contributions that you've helped us learn about is that Alzheimer's disease is a long process, and it begins many years, probably more than 2 decades before we get to the stage of Alzheimer's disease that is so devastating, where people have very significant problems and dementia. And as Dr. Hyman already mentioned, one of the ways we've been able to see this is looking with biomarkers and with imaging. And here are some of the images that I don't know who they are because I stay blinded, but it could be one of you because we are so fortunate to have so many people in the Harvard Aging Brain Study who've contributed by letting us image their brains. So importantly, we can look at the 2 important proteins that change early in Alzheimer's disease with imaging. One is we call amyloid beta, and the other, of course, we call tau, that is in the tangles. The amyloid plaques we know are very high in people who already have Alzheimer's disease dementia. But one of the first things we started to notice, as well as other groups, is that a number of clinically normal people also have very elevated amyloid. And again, as you heard from Mr. Chen, this is really allowed us to detect evidence of disease before people have symptoms, and hopefully one day prevent them from developing problems. But not only can we see evidence of this elevated amyloid, we can also see that people with very high amyloid in their brain also start to show evidence of tau, these tangles spreading throughout the brain. And what we want to do is try to prevent that. So as Dr. Hyman already mentioned, we've had some challenges in clinical trials. And I'll say that as of my last count, we've had 13 phase 3 clinical trial failures at the stage of symptomatic Alzheimer's disease, MCI or dementia. Mostly these have been anti-amyloid trials, but they're really starting, most of these trials have started later. And I put an asterisk here because later next week, we'll learn more about what the FDA thinks of one treatment that maybe showed some signal at this stage of disease. Again, as I mentioned, these changes begin in the brain years before people get to symptoms. And even by the stage of MCI, there's evidence of some irreversible brain damage already. This is not so surprising because this is exactly what happens in many chronic diseases. Think about cancer or stroke or HIV or osteoporosis, and what happens if you wait to go to your doctor until after symptoms are apparent. So we want to detect and treat disease earlier. The problem is the earlier you go, the harder it gets to identify people who will progress and then track the impact of a drug that you're going to try in that population. So this leads to the idea of trying to go earlier in trials. What we've done primarily has been, I'll say, tertiary prevention or treatment, taking people who already have symptoms and trying to slow their progression. And this is very important. And again, maybe there's some evidence that if we go aggressively enough at that stage of disease, that we might be able to slow progression at least a little bit. But we all want to go earlier. We want to prevent people from getting to that stage of disease. We all want primary prevention. I like to say we're going to find something that will put in the drinking water of everybody over the age of 18. We're not there yet. So we're really at this phase of secondary prevention, where people have changes in their brain, they don't yet have symptoms, and we're going to try to keep them from progressing. Now, most of what is going on in clinical trials right now and over the past 5 to 10 years has really been this idea of helping the brain clear the amyloid plaque. And I do think it's very evident now that we have multiple drugs, mostly antibodies, that's proteins that attach to the plaque and help the body clear this. That we have now drugs that can get amyloid out of the brain. And what we hope is that this will be associated with slowing of cognitive decline and preventing ultimately dementia, or at least preventing progression. But, and I apologize for all the data on this slide, but I just wanted to show you some of those messy actual lines, as Dr. Hyman did, of what it looks like. We realize now that amyloid and tau are both critical for causing dementia or being associated with dementia of Alzheimer's disease. But we hope that we could potentially try to prevent, especially the tau tangles, from spreading the brain by going early enough. As you can see up here, these shooting up arrows are people that once they get to a stage of elevated amyloid in their brain that goes above a certain level, that seems to be associated with this kind of explosion of tau in the brain. And we call this the catastrophe. And most recently, we've been able to see that that's happening actually over a pretty short period of time, this tau increase, and associated, unfortunately, with people who move to mild cognitive impairment or dementia. All of the people in this upper graph all started out normal, without any cognitive problems. But over time, as their amyloid increased, unfortunately, their tau also increased. And as you can see in this bottom part here, we're now able to track how much tau accumulates every year in these studies. And that will allow us to test better whether our drugs are really making a difference. So I'll talk just very briefly about the A4 study, which was already introduced. This is the first of its kind, a secondary prevention study in people aged 65 to 85. And some of you on this call have been in this study and are still in this study, and I'm so grateful. And we have 67 sites around the world. This again is a phase 3 trial, which means if it's successful, we might be able to bring this as a preventative treatment to people. We did make some major adjustments to this study based on other evidence. We quadrupled the dose of the antibody because we've learned that we're going to have to be more aggressive in getting a lot of these antibodies as much as possible in the brain. I won't go into the details of this study, but I do want to show you one of those cartoon diagrams of what we're hoping to do. So here at the top is what people who don't have elevated amyloid are like. They have some decline, but it's slower. And in red, unfortunately, is what we've learned from the Harvard Aging Brain Study and ADNI and many other studies, is what happens to the cognition of people who have elevated amyloid, even if they're still normal in the beginning. We hope that with an anti-amyloid treatment, if we start early enough, we can bend this curve and actually bring people closer to the low amyloid group's trajectory. And this is the idea in the A4 study. So here are some results that we presented and published earlier this year, just to show you, and those of you who have contributed to this. So we had to screen about 15,000 individuals. We really do need every volunteer, every participant to help us. And in the clinics at these 67 sites, we screened 6,700 people. We got PET scans on 4,486. We had predicted that about 30% of individuals would have elevated amyloid in their brains, and it turned out to be 29.5%. So that prediction was right on. And then if people had elevated amyloid, they were able to continue in screening and ultimately get randomized to either getting solanezumab, the antibody in this first study, or placebo. And we also are following people, and some of you on this webinar might be in the LEARN study, who didn't show elevated amyloid, because it's very important to follow people who don't have that risk factor to see what happens. And as Dr. Hyman said, to understand more about why some people decline faster or slower, and ultimately what makes them resilient. So I'll just show you a tiny bit of data, because this one really surprised me a bit. So when people came in to screen for the A4 study, we asked them a questionnaire, a very simple questionnaire about whether they had experienced any changes in their memory or ability to do things over the past year. All of these people are still normal, but we said, have you had any subtle changes? And this was done 1 month before the PET scan. So the PET scans really did not bias anybody's opinion here. And what we could see is those who had elevated amyloid here in red, the participants themselves, so they were much more likely to have experienced some subtle changes, such as difficulty using their iPhone, like I had this morning. The study partner also said that there were a little bit more changes, although this was not as apparent as the person themselves. Now, this made me realize that when people say they're experiencing these subtle changes, if they're really a difference from the year before, we have to pay attention, because it is more likely that they might have these changes in the brain consistent with early Alzheimer's disease. But of course, we need to understand, because lots of us have very subtle changes, and not all subtle changes with aging are Alzheimer's disease. And a lot of what we will do with this work is try to help differentiate what you should be worried about and hopefully what you shouldn't be worried about. So my last minute here, I'm going to talk about our newest study, which we actually randomized our very first participant, I think down in Rhode Island, about a month ago. And this is called the AHEAD study. And this study even pushes the envelope more than we did in A4. We're going down to age 55 now, although people who are 55 to 64 have to have additional risk factors, such as a genetic risk factor or a strong family history. The other thing in this study is we're using personalized dosing. So people who have lower levels of amyloid that we call intermediate amyloid are going to come into this A3 study. And here we're really trying to prevent them from accumulating the amyloid altogether, and we'll be looking at tau. If people screen and they have very elevated levels of amyloid, again, everybody's normal in this study, they will go into what we call the A45 study. And there we have people who will get pretty aggressive dosing in the first 2 years to try to knock down that amyloid plaque as quickly as we can. And then we'll have them on maintenance dosing of an antibody for the next 2 years. So I'm very excited that this study has finally launched, and hopefully we'll have more data from that by our next participant visit. So just to end, I want to say that I'm so appreciative of everybody who's helping us, both in the observational studies, which make these trials possible, and the continued data from the Harvard Aging Brain study and from the memory study in MADRC really are helping us to understand how to design and analyze these trials. We do have what's called a trial-ready cohort, NIH-funded study that's using a web study to help us find people who might be appropriate to come in and screen. We're working very hard on blood tests to detect early Alzheimer's disease, have a new finding on this that's being published next month, and really helps us maybe get to blood tests that'll help us decide who should get a PET scan or spinal fluid. As you heard, we're doing smartphone testing and iPad testing. And very excitingly, we're about to start planning an anti-tau trial, which I think will be really important. And especially for people with symptoms, I think eventually we're going to need a combination of amyloid and tau. I do want to talk about the importance of diversity in our trials. This is something we've worked hard on, but we haven't fully succeeded. We're already learning from the A4 screening data, as Dr. Hyman said, that there are differences by gender, by sex, by race and ethnicity, and this might be really important in how we develop the best treatment for every person. I'll also say that COVID-19 is definitely a challenge for our trials, but they are up and running again, as you'll hear from Dr. Marshall. And it's hard. We want to keep everybody safe, but we also feel that Alzheimer prevention remains an urgent issue that cannot wait. So here's the Harvard Aging Brain team, and you saw lots of pictures before, and I just want to again say thank you so much. We definitely cannot do this without you, and I can't wait. I think that 5-year prediction might be right on, and I can't wait to have the celebration with all of you, hopefully before that 5 years. Thank you so much.
Thank you, Risa, so much for being actually a leader in giving hope. You know, what prevention does is it helps us realize that maybe we have some control over our actions, our behavior, what we can do to help delay the disease or fight against the disease. So thank you so much. And Godspeed in going to take care of your mom. We do appreciate you taking the time to be here with us today.
Thank you so much.
So now we're going to move to a distinguished panel of people doing the research. The leader of the panel is going to be Dr. Brad Dickerson. Dr. Dickerson is a professor of neurology at Harvard Medical School and the director of the frontotemporal dementia unit. He is a leader of the LEAD study looking at imaging and early onset Alzheimer's disease, and overall, a wonderful guy. So Brad, thank you so much for leading this panel. All of you who are on the panel, please turn on your videos, and I'm going to turn mine off. So Brad, it's all yours.
Thank you very much, Doreen. And thanks to you and Judy and everyone for organizing this. I think we all share the feelings that we wish we could be in the same room together, but like Brad Hyman said, I think we wouldn't all fit if all of us on this Zoom were in one auditorium. So it's nice to be able to reach people that might not ordinarily have been able to attend. And I think what is probably apparent to everyone listening is that we have an amazing extended team here. This is a team that all works together of people that are dedicated with a lot of passion, as you're hearing, to the mission that we're all aiming to achieve. And we all agree that we can't do this without you, but I want to say that we all agree that we can't do this without each other. And it's wonderful to be part of such a collegial team of experts in so many different areas that I think are really, is an unparalleled team in my view, worldwide. So I'm going to moderate a series of short talks where a number of members of our team are going to highlight in about 4 minutes or so, a little speed dating talk here about what they're doing and what they're excited about in their research. And we'll go from one to the next. And then after that's finished, we'll have time to take questions and have some discussion. So first, we're going to start with Dr. Steve Gompertz, who is an assistant professor of neurology at Harvard Medical School and director of the Massachusetts General Hospital Lewy Body Dementia Center of Excellence. So Dr. Gompertz, please start us off.
Well, thanks, Brad. First, I just want to thank you all for participating in our research efforts and joining us today. We really do appreciate all that you do to help. My clinical research program is focused primarily on dementia with Lewy bodies and Parkinson's. These are common diseases afflicting many people, including some well-known people like Robin Williams and Bill Buckner. But despite this, I think they've been understudied in DLB in particular, and we and our colleagues are working hard to rectify that. I also study the other so-called atypical Parkinson's syndromes, such as PSP and cortical basal, often collaboratively with Brad Dickerson. Our work is highly translational, trying to understand how these diseases work so that we can develop novel therapies against them. And to this end, we have a clinical research program and a bench research program, and I'll emphasize the clinical work here. One major clinical research program is focused on using brain imaging to better understand these diseases. For this purpose, we use PET molecular imaging as well as MRI. As you've heard from Risa and from Brad, PET imaging allows us to examine specific molecules in the brains of patients. One of our central projects has been to use this technology to understand whether and how Alzheimer brain changes of amyloid and tau may contribute to the development of thinking and moving problems in DLB and Parkinson's. And our findings to date show that beta amyloid is quite common, but certainly not universal in both DLB and Parkinson's, and is not harmless. When present, amyloid seems to accelerate cognitive decline over time and is also associated with exacerbated thinning of gray matter in specific regions of the brain. In addition, tau, when present, also appears to impact thinking in these diseases. This work is very much ongoing and an active effort. That said, the findings to date do raise the possibility that drugs developed for Alzheimer's targeting beta amyloid and perhaps tau may also help in patients with DLB and Parkinson's. Another project underway builds on the observation that master regulators of gene expression called HDACs change in DLB and Parkinson's. HDAC stands for histone deacetylase. One of these, a molecule called HDAC6, actually colocalizes with Lewy bodies, the key change in the brain that occurs in DLB and Parkinson's. We've been interested in the possibility that HDAC changes might be an early abnormality in people with these diseases and might serve as a useful marker for them, perhaps guiding new therapies targeting HDACs. To test this hypothesis, and for this reason, we've teamed up with investigators here at Mass General at the Martino Center, who've developed a PET tracer called MartinoStat that labels HDACs in living people. Using this tracer, we're imaging HDAC changes in people with DLB and Parkinson's. I think Dr. Anna Goodhart, who's been a critical part of this effort, may tell you a little more about this shortly. On the clinical end, we've also just begun to participate in clinical trials for DLB, very much in collaboration working with the terrific infrastructure of the Alzheimer's Clinical and Translational Research Unit. Specifically, we led the MGH effort in a recent clinical trial for DLB called ASCEND-LB, focused on determining whether a drug called neflamapimod can improve thinking in DLB. This was a phase 2 clinical trial that I or Dr. Arnold can tell you more about, but the exciting news is that the data analyses have been completed and the company has very recently reported that the drug seems to work, improving thinking on a battery of cognitive tests compared to placebo. The study will be presented next month at a clinical trials conference called CTAD. And I think the results set the stage for a definitive phase 3 study that, if successful, might mean a new medication to treat patients with DLB. So I'm really excited to see clinical trials develop for DLB, this effectively disease treated as an orphan disease for far too long. And I think we're really in a good place to make some dents. We also have several research efforts going on at the bench and in animal models, including efforts to develop a PET imaging tracer to label Lewy bodies for DLB and PD, a long-sought goal and an elusive one to date, as well as efforts to understand and rescue failure of the brain circuits affected in these diseases and in Alzheimer's, as well as efforts to develop new treatments for DLB and Parkinson's by attempting to exploit environmental exposures that actually seem to reduce the risk of Parkinson's disease. And I guess I'll stop there, and I just want to thank you all again. As research participants in the MADRC, you enable all of our human studies, and along with our patients, you motivate all of the work that we do.
Thank you, Steve. It's exciting to hear about the progress that's being made in your work on dementia with Lewy bodies and related illnesses. Thank you for presenting that. Next, we're going to turn to Kate Papp, who is a clinical neuropsychologist at Brigham and Women's Hospital and Massachusetts General Hospital. She's an assistant professor in neurology at Harvard Medical School, and we look forward to hearing about the work that you've been doing, Kate. Thank you.
Thank you, Dr. Dickerson. So as a clinical neuropsychologist, I studied the changes in memory and thinking that occur in Alzheimer's disease and related dementias. And many of you as research participants have taken the cognitive tests that we use to assess memory and thinking. These are tests that require you to learn and recall short stories, to repeat increasingly long lists of numbers and letters, and to do tasks like copy figures. Right now, these tests are one of the primary things that we use to determine whether a medication or an intervention is successful. Everyone takes these tests in a trial, and then we hope that the people who are on the medication show less decline on these tests or measures over time. But as we know, there have been a lot of failures in Alzheimer's disease clinical trials, and there's been a movement towards earlier intervention and treatment, so trying to prevent people from declining. With that, we also need memory and thinking tests that are more sensitive, tests that can detect more subtle changes more reliably, as well as tests that can assess changes in thinking over much shorter time intervals. So moving from years to weeks to months. So I've been working on a very exciting project that's funded in part by the MADRC, as well as under the direction of the Harvard Aging Brain Study. We've developed cognitive tests that can be completed on your own smartphone. You get texted or emailed a link, and then you spend about 15 minutes doing a test on your phone. So in particular, we're interested in looking at people's learning curves when they complete the tasks every day for 5 to 7 days. We have some really exciting preliminary data that suggests that people who have signs of Alzheimer's disease in their brains, but no other frank memory problems, show slightly worse learning curves compared with their peers. So what we hope is that measures like this can eventually be used in clinical trials to reduce the amount of time that it takes to observe the effects of a medication or intervention. So I want to thank those of you who have already been our vanguard participants in piloting this remote assessment. Your feedback has really been invaluable, and it's critical for us to make progress in developing these more sensitive tools. That's it. Thank you.
Thank you very much, Kate. It's really exciting to hear more about this work, which I know you'd been doing before COVID, but which we all recognize is even more important and fundamental now that we have to do so many of our assessments remotely.
Absolutely.
Great, thanks. So next we're going to turn to Jaquel Quiroz, an associate professor in the Department of Psychiatry and Neurology at Massachusetts General Hospital and Harvard Medical School. Dr. Quiroz currently serves as director of the MGH Familial Dementia Neuroimaging Lab and Multicultural Alzheimer's Prevention Program. Please tell us about what you've been up to in your exciting work lately, Jaquel.
Thank you, Dr. Dickerson. And I want to start also by thanking all our research participants, as it has been saying before. So our research is only possible because of the support and dedication of participants. So big thank you, and also muchas gracias. I'm very interested in understanding aging, mild cognitive impairment and dementia, in multicultural and diverse individuals. As Dr. Dickerson mentioned, so I'm the director of the Multicultural Alzheimer's Prevention Program, or MAP. And as part of this program that is also affiliated with the MADRC, we have now a few studies looking at aging in older Latinos, and older Latinos because it's the first one that I got the opportunity to connect since I'm Latina myself. So one study is as part of the Harvard Aging Brain study, and it's known as the Latino HAPS. And we are now putting together that cohort within the HAPS study. And also there is another study that we are doing with the community at large that is now known as the Boston Latino Aging Study, or BLAST. So as part of this new study, so we're going to be recruiting individuals that are older than 60 that have a background from any country in Latin America. And the goal is to try to understand how the disease or even cognitive decline or a normal aging appears in Latinos compared to other groups. So we also started recently a study with bilingual individuals. And the main goal is, with this study, is to understand how memory, and in particular, how language decline actually presents in bilingual individuals as they go through mild cognitive decline or impairment, and also as they go through aphasia, primary progressive aphasia or dementia. So the same, like I started with bilingual individuals, English, Spanish first, but the goal is to actually extend it to be able to study bilingual individuals or multilingual individuals with other languages as well. So in the past few months, and the same in the context of COVID, so I also started a study looking at the impact of COVID-19 on memory function, and in particular, the risk to memory decline and Alzheimer's disease. So we have been collecting data. First, we started with online data collection, completing a survey. So far, we have more than 2,000 people that have completed the survey. But now we are going to move to trying to characterize some of these individuals that participated in the survey and inviting them to complete some remote assessments to get a better assessment of their cognition. And the same, like we're interested in seeing how COVID can impact the risk, but also like what are the possible protective factors playing a role there as well. And the second area of interest of mine is autosomal dominant Alzheimer's disease. So this is a form of the disease that is explained by genetic mutations in 3 genes, like presenilin-1, presenilin-2, and the APP. And these genes usually lead to young onset dementia, usually before the age of 50. So I have been studying a large Colombian family with these mutations, and with the goal to characterize and understanding preclinical Alzheimer's disease. And most recently, I have been studying individuals from these families who remain cognitively unimpaired for several decades, despite having this genetic mutation to develop early onset dementia. So we are now studying these outliers to see if we can identify some potential protective genetic factors that can help us understand protection against dementia. Thank you very much. That's it. Thank you, Brad.
Thanks, Jaquel. It's really an exciting set of distinct but interrelated lines of research you're working on, and it's really exciting to hear about some of the hot topics in understanding COVID impact on memory and thinking about ways to better represent our community in the studies that we do. So thank you for summarizing that. Next, we're going to turn to Gad Marshall, who is an associate professor of neurology at Harvard Medical School and medical director of the Brigham Center for Alzheimer's Research and Treatment. Look forward to hearing what you have to say, Gad, about clinical trials and related activities. Thank you.
Thanks, Brad, and thanks everybody for joining and for your ongoing participation and support in our battle against Alzheimer's disease and related dementias. As Brad said, one of my roles is running the day-to-day operations of the Center for Alzheimer Research and Treatment at Brigham, or CART, where we run primarily clinical trials. These are multi-center studies, and we also run some multi-center imaging studies. And so currently, for example, we're recruiting participants for the AHEAD prevention trial that Dr. Sperling mentioned earlier. We're also recruiting for the CLARITY trial, which uses the same drug that's currently has a alphanumeric code BAN2401 that is an antibody against amyloid that's meant to reduce amyloid plaque buildup in the brain. It uses the same drug as the AHEAD trial. However, the CLARITY trial is focused on individuals with early symptoms at the stage of mild cognitive impairment and to a lesser extent in the stage of mild dementia. And so those are 2 examples of clinical trials that we have that are currently recruiting participants. We have several other trials that are ongoing that finished recruiting participants, several of which are actually focused on the tau protein that was mentioned. These are antibodies against the tau protein, trying to prevent the abnormal form of tau from propagating from nerve cell to the next nerve cell. We also have some observational studies, such as TrackPad or the trial-ready cohort that Dr. Sperling mentioned, that is meant to build a cohort of participants interested in clinical trials and then recruit them for trials when they're available, as well as the Alzheimer's Disease Neuroimaging Initiative, or ADNI, that's been going on for 16 plus years across North America, and recruits participants ranging from normal to mild dementia, and have them undergo multiple imaging assessments. And so we've been an active site for that study for many years. And so that's an example of the type of studies that we have at CART. We have a large team of investigators, nurses, coordinators, project manager, and really try to do the best job in terms of interacting with participants and study partners who put such a big effort and their participation in these studies to get us an answer for whether or not these drugs or interventions can finally help improve our battle against Alzheimer's disease. So that's one role that I have. Another is primarily at MGH, where I focus more on my own research that over the years has had two main areas of interest. Instrumental activities of daily living, or IDLs. In brief, basically high-level daily functioning is an area of interest of mine in terms of how individuals perform on a regular daily basis, and how to assess that, and how to assess the earliest changes in that level of functioning. And so we've been able to develop new instruments to try to get to that answer. Initially, we used the regular phone, and now more recently we switched to a smartphone with a set of tasks that challenge individuals in a way that relates to some medical tasks that they would generally do. And more recently, we've also been able to relate these assessments to imaging with amyloid and tau PET and MRI. And then we also have, I have an interest in neuropsychiatric symptoms, or mood and behavior symptoms that occur in late life that are part of these neurodegenerative diseases like Alzheimer's disease. In particular, apathy or lack of motivation, lack of interest has been an area that I've focused on and related to many different imaging modalities in the brain to try and figure out where this lives in the brain. And this is a very frustrating symptom. Individuals may seem like they can do things, they have the ability, but they don't do them. And it's not necessarily under their control within their initiative, but it could be very frustrating primarily for their loved ones. And trying to understand that and then hopefully translate that into more effective treatments has been an area of focus of mine. And some of the studies we've been running lately at MGH are focused on individuals with cognitive symptoms at the early stages to try and complement the extensive number of participants we have in the Harvard Aging Brain Study that focuses on individuals who start off without cognitive symptoms and age. And so we try to complement some of that data with data on individuals with symptoms and merge those studies to get even more answers. And so I'll stop there, and I'd like to, again, thank all our participants and study partners for putting up with all our assessments and tests over the years. Thank you.
Thank you, Gad. It's really exciting to hear the number of lines of research that include what I think you would probably call basic imaging cognitive neuroscience efforts to understand why some people are so motivated, like hopefully all of us feel today, to be part of this, but others, unfortunately, many of our patients really suffer from that challenge, as you said, and the clinical trials that you're leading that hopefully will test the next generation of treatment. So thanks for that brief summary. Next, we're going to turn to Dr. Matthew Frosch, who's the Lawrence J. Henderson Associate Professor of Pathology at Harvard Medical School and Director of the Neuropathology Service and Brain Bank at Massachusetts General Hospital.
Thank you. Thank you, Brad. And I just want to thank all the participants who've come to listen to us this morning. I think it's really very motivating for us to know that you are there and want to know what we are doing, and it really does help us focus on this significant problem that affects all of us. I know that many of you during study visits have had a conversation about the possibility of brain donation. And when people agree to that and when the time comes, that's where I come into play, not just for the MADRC, but for most of the neurodegenerative disease research groups that operate across the Harvard system. And so what I want to do is tell you a little bit about what we contribute both for you and how to research. And I think for you as participants, families who are affected by the disease, our ability to both provide a diagnostic report, which establishes a diagnosis, and as Dr. Hyman mentioned, really highlights what is unique about the way the disease and other things that accumulate with age have affected the brain in an individual. That individual, that personalized medicine aspect of the neurodegenerative diseases like Alzheimer's disease, like Lewy body disease, like the frontotemporal dementias, really helps, I think, clarify what was unique for any individual. That turns around, and as we begin now to try, as we are making a major effort to focus on individuals who've been in clinical trials, to really be able to illuminate why trials may not have worked, not because the drug was not right, not because the drug was not acting right, but because somebody's underlying dementia is driven by multiple different components, and that our trial is only effectively treating one part of it. So that interplay of the diagnostic autopsy with the research aspects of clinical trials, ongoing research and data collection, I think is really central to the contributions that I make. I do want to highlight a few other things that have allowed me to work with several people who are up on the screen with you as well, which include Gad talking about subjects from trials that he's been running, with Dr. Gompertz about Lewy body disease, Dr. Gomez-Isla, who you'll hear from in a minute, Dr. Arnold, which is really to understand how the biomarkers, what's done with blood that's drawn, what's done with measurements from cerebrospinal fluid, what's done with the imaging, whether it's an MRI study or a PET study, how do those actually relate to what is happening in the brain? And I think illustratively, the initial tau studies really suggested that the agents might be widely usable in a variety of different diseases. And as we took subjects who came to autopsy who had been imaged recently, and were really able to sort that out, we were better able to define how that tau agent needs to be used clinically. And I think that's an enormously important part, sort of closing that loop of discovery for diagnostics. And clearly, as you heard from Dr. Sperling, the diagnostics that become the entry points for clinical trials have to be validated. They are presumptive, but without the validation that, that the tissue-based research brings, it's really very hard to rely on them. And finally, the samples that we collect are distributed not just within the people you see here, but really around the country and around the world. We typically make about 100 tissue distributions a year. All tissue goes out completely de-identified. None of that data, the personal data, leaves, but it comes with the rich data that's been collected from every visit, from every imaging study, and from every blood draw. And that makes it useful for investigators at other institutions, both around this country and in fact around the world where we have collaborations. And we participate in a large number of national and international consortia where other groups working like us in other centers are really pooling what we have in an attempt to gather as much insight as we can. And then I'd like to just highlight, to emphasize comments that Yikel made, clearly, in addition to participation in trials of a diverse group being important, it is really important that we are able to reach out to diverse communities to participate in the tissue donation as well. Because if we don't close the loop for them in the same way, we're not serving them anywhere near as well. So with that, I'll close and pass the microphone to the next.
Thank you, Dr. Frosch. I'm sure it's so valuable for our participants on today's meeting to hear more from you about the multi-pronged value of this critically important and unique donation of a person's brain after death. So thank you for summarizing that. Next, we'll turn to Teresa Gomez Isla, Associate Professor of Neurology at Harvard Medical School and Assistant Director of the Massachusetts Alzheimer's Disease Research Center. Thank you, Teresa.
Thank you, Brad, and thank you, dear research participants. I hope you know that when we say you are our heroes, we really mean it. In the middle of this pandemic that is adding so much suffering to our community and to the world, more than ever, we care about you and about your families. And more than ever, we remain committed to work with you to find better treatments and eventually a cure for our dementia patients. I have to tell you at least a couple of words about Steve Johanson because I had the great privilege of being one of his doctors. Steve and his wife, Judy, forever became part of my life from the very first time they walked into my office. And they taught me lessons that they don't normally teach you in medical school. Steve taught me the true meaning of resilience, generosity, and hope. He would never miss a chance to contribute to our research till the very end, even knowing that it was unlikely that a cure would arrive on time for him as an individual. I think he would be very proud to see all of us continue on with this work. And I think he would also be very pleased to see that the future of our center is in the best possible hands now, with a new, young and bright generation of future leaders. One of the most rewarding aspects of my role as Memory Division Chief and Clinical Core Leader is to help training some of the very best and bright young clinical and research fellows. And I think you are gonna be delighted to meet two of them, Dr. Anna Goodhart and Dr. John Dixon. And I'm sure you'll understand why we are convinced that the future is bright. Dr. Goodhart?
Hi, thank you so much everyone for coming to this and for participating in our research. So I work primarily with Dr. Gompertz, who you heard from earlier. I'm interested in how dementia and memory problems overlap with moving problems like Parkinson's disease. So I use PET imaging, so the amyloid imaging and the tau imaging that Dr. Sperling talked about, to study memory and thinking problems in patients who also have moving problems. And I also correlate these imaging findings with some of the neuropathologic findings that Dr. Frosch talked about with the brain donation program. Additionally, I use PET imaging to look at epigenetic molecules that Dr. Gompertz mentioned. So these are the regulators of our genes. And I use this PET imaging technique to better understand how our genes might be aberrantly or differently regulated in people who have moving and thinking problems. So our genes are actually regulated by environmental factors. And so along with that, I'm also studying how smoking might play a role in the development of moving and thinking problems. And so how the environment and genetics might interact. So we really appreciate all of you participating in our research, and thank you for giving me the opportunity.
Dr. Dixon.
Yes, first of all, I wanted to thank you all for participating in the dementia research that's ongoing in our institutions, and also for joining us for this event today. I also wanted to thank you for the opportunity to tell you about some of the research that I'm doing, which is being conducted in Brad Hyman's lab, who you heard from earlier. My research in particular focuses on understanding the role of tau in the development of dementias and how they progress. Some of the previous work from the Hyman lab has shown that in Alzheimer's disease, a protein called tau, which accumulates in neurons, also disrupts the normal transport process of proteins into and out of the nucleus, which is where the DNA is stored. I've been using autopsy tissue from the brain bank that's allowed me to examine other diseases where tau accumulates, such as frontotemporal dementia and other diseases which are known as tauopathies. And through these studies, I found that there was this same disruption of nuclear transport that was seen in Alzheimer's disease in these other tauopathies. And this is an important finding because it suggests that disruption of this nuclear transport process is a general feature of neurodegenerative diseases involving tau. And I think that could be important to know in terms of thinking about therapies for the future. If we're able to target this process, the therapies may have benefit not only for Alzheimer's disease, but for other types of dementia as well. One of the key questions that comes up from understanding this dysfunction of the transport process is which proteins that are transported might be affected. And I've also been using some of the techniques in lab with the autopsy tissue, as well as animal models of disease and cell culture, to understand how this transport process is affecting a particular protein called RNA polymerase II. This is an essential protein that's involved in the process of using the DNA blueprint to make the protein building blocks for the cell. And it needs to be in the nucleus to perform these important functions. But in Alzheimer's disease, I found that this protein accumulates outside the nucleus where it can't perform its normal functions. And this may have a serious impact on the health of neurons in Alzheimer's disease. This is an active area of research that I'm currently investigating now in the lab to try to understand more about why this happens and how it affects cells. But ultimately, I hope that a better understanding of how tau contributes to the development of dementia and its progression might suggest new treatments for these diseases, which I think is what we're all aiming for here. So again, thank you so much for your time and for letting me tell you about my work.
Thank you, Teresa, for allowing Anna and John to share some of the exciting work they're doing under the mentorship of some of our leaders. I think that's one of the things that we talk about all the time, but probably don't have the opportunity to share enough with our attendees today, which is that pretty much every project we work on is not only interdisciplinary, but also offers opportunities for our more senior members of our labs and clinicians to mentor more junior members of our team and grow the next generation. So it's a really wonderful opportunity to talk about that. Thank you. So finally, we're going to end the presentations before we move into a panel discussion period, Q&A period, with a word from Stephen Arnold, Professor of Neurology at Harvard Medical School and Director of the Alzheimer's Clinical and Translational Research Unit at Mass General. Stephen?
Thanks, Brad. Whoops, am I unmuted? Yes. So thanks, Brad, and thank you all, really. So I came to Mass General about 5 years ago from Philadelphia with one postdoc in tow, and to really help grow the clinical trials activity on the MGH campus. And you know, I have to say, you know, starting with 2 people, we're now almost up to 30 people spanning, you know, doctors, nurse practitioners, nurses, an amazing group of research coordinators, molecular biologists, lab technologists. And it's all oriented towards how to take some of the really amazing ideas that have been coming out of the labs at MGH and translate them into potential and test whether they can, some of those ideas can actually help cure or prevent Alzheimer's disease in people. So our focus has been on more on what we would call early phase proof of concept trials. You know, some of you have heard me say, you know, we've got hundreds of successful cures for Alzheimer's disease if you happen to be a mouse. But we don't know whether those are going to work in humans. Many, probably most, will not, because humans are very different from mice. Some of these things, some of these ideas are actually other medicines that are used for, that are already approved for other conditions like arthritis or diabetes or heart failure. Do they work in people with cognitive difficulties or Alzheimer's disease? So our job is really to, and what we try to do in the ACT-TRU, the Alzheimer Clinical and Translational Research Unit, is to try to be clever and design small, efficient studies to kind of test whether there is some promise for these new medicines, even things like natural supplements, exercise interventions, dietary interventions, to see whether they show any signal of possible benefit in 10 people, 20 people. If they do, then we will graduate those interventions onto the larger studies that I think Risa Sperling described, and GAD has been so capable at running. The other thing that we need to do in clinical trials is to, we're going to try something, but how do we actually know whether it's showing a signal? How do we actually measure whether there is some potential benefits? So another big aspect of our work has been to look at biomarkers. What are the proteins or other things that we might be able to measure in blood or in spinal fluid that can tell us whether a new drug is actually affecting amyloid or tau or inflammation or any number of other markers, so that we can see whether medicine offers the potential benefit. So, I think that's that. So we have a laboratory and we run the biomarker core for the Alzheimer's Center, as well as the Harvard Aging Brain Study now. And, we try to develop new lab tests that we can measure in blood or spinal fluid that might be helpful in clinical trials. So, a few of the things that we have going on now, we're looking at one natural product, a supplement called nicotinamide riboside, to see whether this is something that helps mitochondria in the brain and in the body. So the way that brain metabolizes energy. And so we've designed a clinical trial to actually test that. We're looking at another commonly used medicine, Concerta, which is a stimulant medicine with brain training games to see if it'll help the symptoms of people with mild cognitive impairment. And we've designed that in a very kind of an interesting way, where we do multiple crossovers in the design to see whether the medicine actually helps the individual person. And I can perhaps explain that more a little bit later. We've got 2 anti-tau trials that are going on. We're about to launch a trial of a anti-tuberculosis vaccine that's been given to hundreds of millions of people over, around the world for decades now, because there's some indication that it actually might help prevent Alzheimer's disease. Does it? How do we design a trial to figure that out? How do we roll it into a clinical trial to test it? Who is it that we want to recruit into this trial? These are the things that we kind of think about and help design. So, there's an awful lot going on. This has been, I as a newcomer really to the community of Alzheimer research here at Mass General and Brigham, it's just been an unbelievably rich environment with amazing people to work with in order to really make a difference. And I think as much as I appreciate my colleagues, I think, we all appreciate you, who participate in the studies, because we really do view the work together as a partnership in order to bring a cure and a prevention to Alzheimer's disease. So I'll stop at that point and we can go into the panel discussion.
Thank you, Stephen. It's wonderful to hear some specifics about the different kinds of treatments that you're trying. So we have some questions from the audience, and as is often the case in these kinds of discussions, there are a number of questions about potential modifiable risk factors or conditions that people have heard about. Let's start with sleep. I wonder maybe if Steve Gompertz, you might want to just mention, people often ask about how sleep has a role in neurodegenerative diseases, and maybe you could touch briefly on disorders of sleep, and then maybe you or others might want to just discuss briefly some of the interesting work that's been going on the interactions between sleep processes and Alzheimer's pathology.
Sure, I'm delighted to take a crack at it, and other folks are certainly welcome to chime in too. We've known for some time that there are some risk factors for neurodegenerative diseases that manifest as changes in sleep. And a classic one known as REM sleep behavioral disorder shows up as people acting out their dreams, so-called dream enactment. So late in the stage of sleep, people will go transfer from slow wave sleep to what's called REM sleep, where our bodies are paralyzed and our eyes go back and forth horizontally. But in people at risk for Parkinson's or dementia with Lewy bodies or another disease called multiple systems atrophy, one will lose that normal paralysis of REM sleep and start to act out our dreams. So this REM sleep behavioral disorder can be confirmed on a sleep study, and it's now being used as a biomarker to identify people at risk for these diseases I've mentioned, such as DLB and Parkinson's. And it's turning out to be a pretty interesting way to recruit a really enriched group of at-risk people to see if we can prevent the onset of a synucleinopathy, Parkinson's, dementia with Lewy bodies, or multiple systems atrophy. There's been a lot of work afoot in the basic science work, particularly in animal models of disease looking at sleep, showing that, really interestingly, in animal models of Alzheimer's disease, amyloid is cleared in sleep, particularly in this phase of sleep called slow wave sleep. That's still a very active area of research. There's some data to suggest that sleep disruptions in people also manifest as an increase of amyloid accumulation. And there is a lot of work afoot working to see if we can link improving sleep to reducing our risk for certainly developing symptoms of neurodegenerative diseases like Alzheimer's disease. In our own work, we find that this is the electrophysiology stuff I touched upon earlier, that brain circuit failure manifests particularly in slow wave sleep compared to other phases of sleep or wakefulness in the hippocampus in models of Alzheimer's disease in which amyloid is particularly present. And we're trying to understand that and see if we can't focus therapies particularly to that stage of sleep and see if we can perhaps reduce amyloid and perhaps improve symptoms in so doing.
Great, thanks, Steve. I think if we want to cover the many questions, I think we probably don't have time to get more into sleep, even though I know a number of us have studies ongoing that are looking at this. So let's move in maybe to what we might talk about as lifestyle. There were some questions about diet and exercise. And I know that in the HABS study, there's work ongoing in that. I'll just mention before maybe I ask Kate or Gad to talk a little bit about diet or exercise or other lifestyle factors being studied in HABS or other studies. We did a study as part of our multi-center genetic frontotemporal degeneration consortium, where we looked at people who were carriers of genetic mutations like Jaquel talked about that lead to Alzheimer's, but in this case, these mutations lead to frontotemporal dementia. And it turned out that, at least retrospectively, when we looked at differences in rates of progression from an asymptomatic to a symptomatic state, and within people who had symptoms due to these FTD genetic mutations, sort of like what Brad Hyman talked about, there was a slower rate of progression in people that had a more active lifestyle and reported on that in terms of self-report. So I think there's a growing body of evidence from our own work and others' work that these recommendations that we often make at during our clinic appointments about active lifestyles, whether they're physical, social, or intellectual, there's a growing amount of evidence to show that there really is an impact of that on changing rate of progression. So Kate or Gad, do you want to just spend a few minutes talking about how HABS is approaching that?
I'll defer to Kate. I can talk about clinical trials afterwards.
Sure. Yeah. So we are looking at some of these variables within the Harvard Aging Brain Study. So we have a sub-study that involves looking at sleep, but we obviously also collect a lot of information about things like lifestyle factors, like a level of intellectual social engagement, people's social networks, and then asking about things like depression and mood. And we also have a sub-study where we have folks wear a pedometer where we can look at their steps. And we have found that individuals who have sort of higher physical activity or more steps actually show slower decline. And it could be that there's multiple factors that could influence this. So for example, we know that a lot of cerebrovascular risk factors like high blood pressure, high cholesterol are also risk factors for Alzheimer's disease. And by more exercise, you could be sort of helping with some of those risk factors. It could also be that exercise helps with mood, and that could be another factor as well, because we know mood has an impact on cognition. It also could be that the people who walk more in the studies are maybe generally healthier, and there's other variables that we're not necessarily taking into account that make them sort of more hardy against changes in the brain. We also, I think one of the most promising lifestyle interventions is really physical exercise that has been shown from mice to humans to have a protective effect on brain functioning. And there's been an effort that's been being run by the Alzheimer's Association, and actually to run a study called the US Pointer Study, which aims to not only involve exercise interventions, but to have a host of different interventions. So to change people's exercise, to have them meet more regularly with their doctor, to manage their blood pressure and their cholesterol, to have them involved in more social interactions, learning about what's healthy brain aging, what's not, and then also to follow what's called the MIND diet, which is a heart healthy diet. And if we can combine actually all of these lifestyle interventions together, can we even have bigger effects than just exercise alone? So that's a trial that's ongoing. There is a site in Rhode Island, but there is not currently a site here in the Boston area. But that is one way that the research field is trying at a large scale to see if we can take some of these lifestyle interventions and make meaningful changes in people's lives. And then that could potentially influence sort of policy or reimbursement and how these types of interventions are implemented in the larger community.
Maybe I'll just add a little bit. That was great, Kate, that the pointer trial followed the finger trial. You notice a pattern here of digits. That was from Finland several years ago. That was a 2-year multimodal intervention, very similar to what Kate described for pointer, that showed some benefit in cognition in terms of preventing cognitive decline in older adults. And so that was encouraging. And as Kate said, Steve Salloway out of Butler in Rhode Island is leading one of the large sites that's nearest to us in Boston. On a smaller scale, Seth Gale, one of our neurologists at Brigham at CART, is running a smaller study where he has a life coach type of approach that's actually automated with computer prompts to help individuals try and maintain a healthy lifestyle and hopes of preventing cognitive decline.
Thanks, Kate and Gad. It's exciting to see more of these interventions really being evaluated with clear evidence-based outcomes in mind. I wonder, there's a question about lumbar punctures. I wonder, Stephen, if you might want to just answer the question for people that might be on the fence, not sure if they really want to do what you were thanking people for doing in terms of volunteering for a lumbar puncture when they don't really need it, but they might want to contribute in every way possible to research. Can you say a little bit more about how people might want to think about making that decision?
Yeah, so we actually have a study now called the Lifespan Study, where we're trying to, as well as we use spinal fluid testing very, very commonly in a number of studies. I think as Brad Hyman mentioned, spinal fluid itself is sort of the fluid that bathes the brain. And so, you know, I think that we learn so much by testing spinal fluid and measuring all the different proteins, amyloid, tau, different forms of amyloid and tau, as well as inflammatory markers and metabolic markers. We learn so much that, you know, about the health of the brain. And I think also to kind of pick up on some of the point that Dr. Hyman made earlier, you know, people have different types of Alzheimer's disease. There are many different drivers. It's heterogeneous. For one person, it may be inflammation. For another, it's predominantly amyloid or tau. For another person, it's something else. How are we supposed to profile these things and kind of identify and try to match the right person, the right type of dementia and Alzheimer's disease with the right particular treatment or recommendations? So that's done through spinal fluid testing. You know, in some places, like in Northern Europe, spinal fluid testing is part of the routine evaluation of anyone presenting with a memory problem. You know, kind of like a blood test. You also get a spinal fluid test. We have different attitudes in different regions of the world about it. I like to say that it's, you know, a little bit more involved than a blood test, but really not that much more involved. It takes about 20 minutes. It's generally extremely well tolerated. And the amount that we learn from it is really extraordinary. So I'm, you know, a strong advocate for its value. And I think, you know, if people are deciding whether to participate in a research study that asks them about spinal fluid, you know, the thing that I've noticed is that people who have already had a spinal fluid test, or even something like an epidural when during delivery, you know, if they've had that type of thing before, they really have no problem getting another one or others. So, you know, there's more kind of fear and stigma about it than there is really reality. So it is an important aspect of our research. And I just, you know, I'm happy to take individual questions about it too, if anyone's thinking about it.
Thank you, Stephen. We have so many great questions and such a wonderful panel, but I'm afraid we're out of time. I think most of us would be happy to field questions through email if people want to direct those to any of us or to any of the coordinators that are listed on the website. We're happy to follow up afterwards because we really want to answer any questions you have, and we appreciate you being so engaged in the program. So I'm going to turn it back over to Doreen, I think, to finish us up today. Thank you.
Thank you, Brad. And thank you to all our distinguished panelists for sharing your research, for answering questions, and for spending the time. You're a value to us, and as somebody said before, it's wonderful to work with this team. You know, I want to ask Judy to join us again, because we have a final research participant who's going to share some thoughts with us as well. He too is a special guest. And Judy, do you want to introduce him so that he can share his thoughts with us too?
I would love to, Doreen. It's an absolute pleasure to introduce our dear friend and research participant, Mike Belleville. Mike and Steve were great friends who bonded over their dementia diagnoses. They enjoyed a good laugh, a cold beer, and became brothers in arms in this war on dementia. And let's hear more from Mike.
Hello, and thank you all for giving me the honor of speaking today. Just a little bit about myself. I have been married now for 39 years to my beautiful wife, Cheryl. We have 3 children and 6 grandchildren. My career was in IT telephony, working for a large telecom company for nearly 20 years. And I was also a member of the Rhode Island Air National Guard for 12 years. But at the age of 52, I was diagnosed with younger onset Alzheimer's disease. And 3 years later, that diagnosis was changed to Lewy body dementia with Parkinsonism. I am now 59. But let's get to the topic of why we're all here today, and that's research. I have to start off with one of my favorite quotes, and it's by Jackie Robinson. A life is not important except for the impact that it has on other lives. I cannot think of a better person who exemplifies that quote more than my dear friend Steve Johanson. Steve didn't just talk the talk, he walked the walk. He did it with a grace and love unmatched by anyone that I have ever met during my journey. Steve's desire to be a part of something bigger than himself and his involvement in clinical trials is where I learned the importance of getting involved myself. He was a gentle, walking giant, always, and I do mean always, putting other people before himself, even after his diagnosis. When he could not communicate in the way he could before, he still found ways to tell me what I needed to hear, a lot of times without even using words. A gesture here or there, and that grin of his would remind me that this is bigger than all of us. He would give me a kick in the pants, in the nicest ways, of course, when I needed it the most. I was blessed and extremely fortunate to call Steve my friend. And as I like to tell Judy, even though he is not physically with us anymore, he is still teaching people in countless ways. He gave the biggest gift he could give when he donated his brain. To think of how researchers and students are learning from that alone is staggering. I was also fortunate recently to get involved in a drug trial myself this past year. And I have to give a tremendous big shout out to Dr. Gompertz and his team, especially Brooke Fortin. Wow, and I do mean just wow. What an incredible team of caring, passionate people. The trial started in person as is typical, as is the typical protocol. But when COVID hit, Dr. Gompertz and his team didn't miss a beat and went into full swing. We switched to virtual meetings for the remainder of the trial until the final in-person meeting that was held at MGH. What I learned from that, as I'm sure they did, is that it opened the door to other potential ways to have clinical trials. I've talked to a number of people with a diagnosis like mine, and the first thing they say is how challenging it is to get involved in a trial. The two biggest obstacles being transportation and distance from the facility. If you are feeling challenged, or you know someone who is struggling to participate in research, please do not give up without a fight. It took me more than 5 years before I was finally able to participate in a trial. At a minimum, please just talk to the research team. They may be able to make better accommodations for you and your family to participate. I guess you could say that this is my call to action. Research is bigger than all of us. Steve knew that. And it's going to take all of us doing our part in whatever way we can. As I mentioned earlier, a life is not important except for the impact that it has on other lives. You all are having that impact right now just by being involved. But please don't stop there. Just tell others how they can get involved as well. To that end, I'd like to just mention to Dr. Marshall and Dr. Papp about the recent study that they were just talking about with health and wellness. And I'd like to volunteer myself to try to get involved in that study. I like to joke with people and tell them that my new goal is to have my stomach shrink faster than my brain. So if they can help me do that, I'd love to get involved. Thank you all for your time right now, and have a great blessed day. Thank you.
Thanks, Mike. And thank you for that call to action. I hope that the words that you heard today will inspire all of you. You are already participating, so we thank you, but we hope that you will tell your friends and family how important it is to participate in research. Maybe you would even consider brain donation. All of this is enormously helpful to us, and we can't thank you enough for your sacrifices. You know, this concludes our research participant event in honor of Stephen D. Johanson. We've recorded it so you can listen to it again with your friends and family. And I hope that sometime soon next year, we'll be able to have this event in person. And we look forward to seeing you someday. If we can turn off this, and also, you know, follow us on the web, on Facebook, on Twitter. We're developing a brand new website so that all the information that you heard today is going to be more easily accessible to you. Again, thank you very much. If we can have everybody let the slides go and everybody turn on their cameras so we can appropriately wave goodbye. Thank you, everybody. We really appreciate it.
Thank you.
Bye-bye.
Thank you.
Thank you.
Bye.
Thank you. Gracias.
Muchas gracias.